Module 1, Inheritance, Genomic Structure. Following the discovery of DNA structure, it was later deduced that DNA resides within the nucleus in a supercoiled form and contains numerous functional elements. The ENCODE project was designed to identify all the coding and non-coding elements in the genome. While it was originally thought that all non-coding DNA sequence in the genome was just junk, the ENCODE project was ultimately able to assign biochemical functions for more than 80% of the genome. In addition to genes, numerous other genomic elements had been identified. Elements identified include promoter and enhancer sequences, non-coding RNAs, regions of histone modification, and DNA variation, and others. Through the efforts of the ENCODE project, almost 400,000 enhancer and 70,000 promoter sequences have been identified throughout the genome. A promoter is a region of DNA that enables the binding of the RNA polymerase transcription factor complex and the initiation of transcription. By looking at the sequence upstream of many genes, consensus sequence has been identified. Two common elements of the consensus promoter include the TATA box, which is located about 10 base pairs upstream of the start site, and the CAT box, which is located about 20 base pairs upstream of the start site. The consensus sequences for these elements are given in this figure. Unlike promoters, enhancer sequences can either be upstream or downstream of a gene. Enhancers can be sequentially very far away, but are brought near a target through DNA folding. Enhancers facilitate transcription through their binding with the transcription factor complex. Enhancers are bound by a coactivator protein complex called P300CBP, and their location can be predicted using an assay called ChIP-seq, which is designed to identify places in the genome that bind to specific proteins. We'll learn more about molecular assays designed to characterize genomic elements in Module 3. Non-coding RNAs are transcribed from DNA but do not encode for proteins. There are many kinds of non-coding RNAs, including transfer RNA, microRNA, silencing RNA, small nuclear RNA, small nucleolar RNA, and long non-coding RNA. Non-coding RNAs contribute to the vast complexity of our genome by allowing for gene regulation to occur in a number of ways. As shown in the figure by Lee et al., non-coding RNAs can participate in gene regulation at the level of transcription, epigenetic regulation, and post-transcriptional regulation. Histones are proteins around which DNA wraps in order to compact the DNA inside the nucleus. Histones have specific structure and a regulated process for both their formation and for their interaction with DNA and with each other. Histones can be altered through various types of chemical modifications, including methylation, acetylation, phosphorylation, ubiquitination, and sumylation. Histone modification is yet another way in which our human genome is regulated. We'll learn more about histones when we talk about epigenetic regulation in Module 9. There are many ways in which our genome is variable, as summarized in this figure, and we'll learn more about these genomic variability in the next module. The videos to follow will give you more insight as to how these various components of our genome act to influence our health and metabolism.